Hello everyone, my name is Maria Mendoza and today we're going to be going over Worksheet 1. Here are some review concepts that I think everyone should know before getting started. We need to cover electronegativity and the associated vocabulary words. You should memorize the amino acid structures and the functional groups. Electronegativity. So first I'm going to go over the vocabulary words. What is electronegativity? Electronegativity is how well an element can attract electrons. Fluorine is the most electronegative atom. You can see it right here. The electronegativity value is 3.98. The noble gases are not electronegative because they have 8 valence electrons and they're happy. Electronegativity increases as you go to the right and up. Electronegativity describes the unequal sharing of electrons. A covalent bond is when electrons are shared equally. A polar covalent bond is when electrons are not shared equally. Ionic bonds are when electrons are transferred, not shared. When you have an electronegativity difference of less than 0.5, this is a non-polar bond. But when the electronegativity difference is 0.5 to 2.0, then you have a polar bond. And when the electronegativity difference is more than 2.0, then you have an ionic bond. So what is ionization energy? This is the energy required to remove an electron. Some atoms can have more than one ionization energies because you can remove more than one electrons. Only remove one at a time. Electron affinity. How much energy is required to add an electron? Electron affinity increases as you go to the right and up. There is an exception as you go from 1A to 2A and from 4A to 5A. The noble gases have a lower electron affinity. Atomic radii. An atomic radii is 1/2 of the distance between the nuclei. Atomic radii increases as you go to the left and down. Okay, so then I have a little example up in the top left-hand corner for polarity. How do you know when a molecule is polar? So all you do is you take the electronegativity values and you subtract them from each other. For example, one that we see a lot is a hydroxyl group. So you have an oxygen atom and you have a hydrogen atom. So the electronegativity value of oxygen is 3.44 and the electronegativity value for hydrogen is 2.20. And all you do is subtract them. So 3.44 minus 2.20, and that equals 1.24. So the electronegativity difference is greater than 0.5. So this molecule is polar. So if the electronegativity difference had been less than 0.5, then the molecule would be nonpolar. But 1.24 is greater than 0.5. So this molecule is polar. When something is polar, it is hydrophilic, which means water-loving, and it dissolves in water. Okay, by the time that you take your first exam, you're going to have all of these amino acid structures memorized. So what I recommend is that you learn 3 per day. You have to know how to draw them and know which category they belong to. For example, are they nonpolar, polar, or charged? You have to know these by heart. So I recommend that you first learn the backbone, which is what every amino acid structure has, which is this amino group, the carboxyl group, the hydrogen group, And then whatever is in green or purple or orange, that's the R group. And that's what you really have to have memorized for each of these amino acids, as well as the categories. Another thing that you should start memorizing right now is the functional groups. You have to have all of these memorized, so I would just recommend that you learn 3 per day, know how to draw them, and be able to identify them. Okay, so problem 1. Because the properties and biological activities of biomolecules are largely determined by their functional groups, it is important to be able to identify them. In the compound below, circle and identify by name each functional group. So this is acetyl coenzyme A, and starting from the left, we have thioester. So this carbonyl with a S is thioester. Then we have amido, so it's a carbonyl but with an NH. And we have another amido group, carbonyl, NH. Then we have a hydroxyl group, which is also an alcohol. Up here we have a phosphoanhydride. Above we have an amino group, this NH2. And then down below we have a phosphoryl. So that's why it's important to memorize all of the functional groups on that table, because you'll be able to identify all of these easily. Problem 2. For the reaction catalyzed by the enzyme hexokinase, glucose plus ATP is converted into glucose 6-phosphate plus ADP. So here on the left you have glucose and ATP. Those are our reactants. On the right, you have glucose 6-phosphate and ADP. Those are our products. And then they tell you that the equilibrium constant is 7.8 times 10 squared. In living cells and living E. coli cells, the ATP concentration is 6.5 millimolar. The ADP concentration is 0.65 millimolar. The glucose concentration is 2 millimolar. And the glucose 6-phosphate concentration is 1 millimolar. So you have 2 reactants and 2 products, and they've given you the concentration of everything. And then they're asking you the question, is the reaction at equilibrium in E. coli? So they're telling you that the equilibrium constant, K equilibrium, is equal to 7.8 times 10 squared. So you have to do the math in living cells, which is this Q equation here. You plug everything in, and then you look at the answers and you compare. So let's go ahead and set up this problem. Okay, Q is equal to the concentration of ADP, and that is 0.65 millimolar, 0.65 millimolar. And for both of these equations, remember that it is always products over reactants. Okay, so 0.65 millimolar times the concentration of glucose 6-phosphate, because that's the other product, and that's 1 millimolar, all over the concentration of glucose, which is 2 millimolar, times the concentration of ATP, which is 6.5 millimolar. And we just do the math. Okay, so that equals 0.05. Okay, so 7.8 times 10 squared is just 7.8 times 10 squared is just moving this decimal over 2 times. So it's equal to 780. So this reaction is not at equilibrium. It's actually really far from equilibrium. So this number tells us that there are a lot more reactants down here than there are products. So if you remember, the reactants are on the left, and then the products are on the right. And since there are a lot more reactants than there are products, remember that they want to reach equilibrium. So if you have a lot more reactants, the reaction is going to tend to go to the right. And because the difference is so high, it's going to go strongly to the right. Problem number 3. Classify the following molecules as hydrophilic, hydrophobic, or amphipathic. Hydrophilic dissolves in water, things that are polar and things that are charged. So here is an example of a hydrophilic molecule. So this is sucrose, and notice that there's an alcohol, another alcohol, another alcohol. There's a lot of alcohols all around this. So this is a hydrophilic molecule. It's water-loving. Remember that alcohol is polar because when you subtract the electronegativity differences, it's more than 0.5. So all of these alcohols make the molecule polar or hydrophilic. Okay, so what is hydrophobic? Things that do not dissolve in water. So hydrocarbon chains, like octane, for example. So here you have carbon bound to hydrogen. So the electronegativity differences for these two are pretty similar, and that's why this is nonpolar or hydrophobic. You also have benzene and ether. These are all hydrophobic or nonpolar. And then what is a molecule that is amphipathic? That's a molecule that has both parts. So if you look here at this fatty acid, you have a hydrocarbon chain that is hydrophobic and nonpolar, and then you have this carboxylic acid, which is oxygen bound to carbon. Now the electronegativity difference here is more than 0.5, so that means that it's polar. And so this molecule has both a hydrophilic and a hydrophobic portion. And the same thing with dodecyl sulfate, which is a detergent. You have sulfur and oxygen. The bonds here are polar due to the electronegativity differences. And then you have your fatty acid tail. And it's the same thing here for a phospholipid. You can see all of this is polar. And then this charged amine is also polar. And then down here, you've got your fatty acid tails, which are nonpolar. And it's that easy. Problem 4. What is the maximum theoretical number of water molecules that this molecule can hydrogen bond with? Here, you're looking for lone pairs and hydrogens that are bound to electronegative atoms, like nitrogen, oxygen, and fluorine. So we'll go ahead and start with the lone pairs. So here is a lone pair. You have hydrogen, and then oxygen, hydrogen. And then here are two lone pairs. I'll just draw them like that. So you have hydrogen, oxygen, hydrogen, and then hydrogen, oxygen, hydrogen. And then here's two more lone pairs. So you have hydrogen, oxygen, hydrogen, hydrogen, oxygen, hydrogen, and then we have another lone pair. So we have hydrogen, oxygen, hydrogen, and then we have another lone pair, oxygen, or no, hydrogen, oxygen, hydrogen. Okay, I think we took care of all the lone pairs. That was 1, 2, 3, 4, 5, 6, 7. 7 lone pairs. So that's 7 water molecules right here for the lone pairs. And then we have to look for hydrogens that are bound to electronegative atoms. So we'll start at the top. And I'll do these in a different color. Let's do purple. So this oxygen at the top is bound to a hydrogen. Oxygen is electronegative, so that hydrogen, oh, that purple is not dark enough, let's use yellow. Okay, so that hydrogen can bond with a water molecule. So oxygen and then 2 hydrogens. Just ignore this right here. Okay, so then the next atom right here is carbon. Let's use purple again. Carbon is not an electronegative atom, so nothing happens with these hydrogens right here. And then coming down the line, we have carbon again. So again, it is not an electronegative atom, so we do not bond this hydrogen to anything. But then we have nitrogen on the left. Nitrogen is an electronegative atom, so its hydrogens will bond to water molecules. So let's go ahead and do that. So we'll do oxygen, we'll bind here, and then we'll draw its hydrogens. Okay, and then the same thing with the other hydrogen. Okay, so we're done with nitrogen, and we'll go back to purple. So we have this carbon here. We already know it's not electronegative. We have oxygen, but we already took care of the lone pairs. And down here, we have oxygen again, and it is bound to a hydrogen. And since oxygen is electronegative, we'll go ahead and bond a water molecule there. Okay, so there's hydrogen, hydrogen. And I think we're done. Okay, so let's go ahead and count those up. We have 1, 2, 3, 4. So that's 4. So how many water molecules? I have 7 plus 4, so that's 11.
Problem 5. Suppose the major solutes in intact lysosomes are potassium chloride. Let's go ahead and underline that. Okay, potassium chloride and sodium chloride. And then these are the concentrations in molarity, which is just moles per liter. So when isolating lysosomes, what concentration of sucrose is required in the extracting solution at room temperature to prevent swelling and lysis? So this problem is really hard. There's a lot going on here, so we have to look at our clues. They want to prevent swelling and lysis. So immediately that tells you that we are talking about this figure down here on the right. So we're talking about the solutes or the solution that is surrounding whatever it is that they're talking about. So a cell in isotonic solution, there is no net water movement. That means that if there's 20 solute molecules inside of the cell or inside of this lysosome, whatever it may be, then there has to be 20 on the outside. The numbers have to be the same so that no water moves in and no water moves out. So this would make it isotonic. When a cell is in a hypertonic solution, that means that there's a lot more molecules or solutes on the outside, and water always follows the solutes. So the water that's inside of the cell will come outside, it will follow the solutes, and the cell will shrink. And then over here, we have a cell in a hypotonic solution. That means that the solution has less solutes than the solutes on the inside. So water follows the solutes, it moves in, and it creates an outward pressure. And that's what these red arrows are. It's outward pressure. And the cell swells, and it may eventually burst. So in our problem, they're talking about that, to prevent swelling and lysis. So in order to prevent swelling and lysis, you would need something that is in an isotonic solution. Okay, so now I'm going to go ahead and read some book notes for you. The number of solutes affect osmolarity, not the mass of the solutes. So the number of solutes are what's important. If there's 20 inside, there has to be 20 outside in order for everything to be isotonic and for the cell to be happy. This is why many monomers are turned into polymers for storage within cells, like polysaccharides, starch, or glycogen. Buffers can use an inert solute like sucrose to protect organelles from lysing. So that's why in our problem, we're using sucrose. Now, when we're talking about osmotic pressure, we are going to be using the Van't Hoff equation, and it is used to calculate osmotic strength. And I know you haven't talked about this in lecture yet, but it's okay. I'm just going to show you how to use the equation. So this little guy means osmotic pressure. I is the number of particles. Let's see. So I by itself is the number of particles. For example, in potassium chloride up here, we have 2 particles. So the way I look at it is, I think about the periodic table of elements. Potassium is its own element and chloride is its own element. So I is just the number of particles. There's 2. C is the concentration of the particles. So the concentration is given in molarity, or moles per liter. So for potassium chloride, the concentration is 0.2 moles per liter. And then if you have multiple ones of these, you calculate them and then you add them. So, but I'll show you how to do that in a minute. R is just the gas constant. So it's 8.315 joules per mole times Kelvin. Temperature has to be in Kelvin. So here they gave it to you as Celsius, but you have to know that 25 Celsius is 298 Kelvin, and that's something that I would commit to memory. If not, you could just convert. Okay, so let's go ahead and start writing the problem down. Since we know that we're looking at an isotonic cell and buffer like ratio, then we know that the osmotic pressure of sucrose is equal, let me see if my pen is better, is equal to the osmotic pressure of the lysosome. No, that's not working. No, okay. Of the lysosome. Okay, so now we set up the problem. Okay, so the osmotic pressure of sucrose is equal wait, is equal to RT. So R and T are numbers that we don't really want to include in our calculations just yet. We want to begin with the I and the C, the number of particles times their concentrations. And we know that whatever numbers we get for the lysosome, then the number of sucrose, we're going to get the exact same number. They have to be equal to each other if the cell is in an isotonic solution. So we're really just going to calculate the numbers for the lysosome. And that's really easy. It's just plug and chug. So this is equal to RT times, so I see for the first particle, potassium chloride, the I, or the number of particles, was 2. Okay, and then that, so I'm going to put brackets here. 2 times, let's see, 2 times, and the concentration was 0.2. So that's all you do, 0.2. And that's in moles per liter. And then you just add plus. And then you take into account the next solute. So sodium chloride again has 2 elements, so that's 2 particles times, and the concentration of that was 0.04. Okay, and then you just do your bracket. Okay, so 2 times 0.2 is equal to 0.4. And then plus 2 times 0.04. Okay, so that's 0.08. Okay, so when you add them together, it equals 0.48. Okay, and then what's cool here is that the R and the T cancels out from the sucrose and the lysosome. So really, you just get 0.48 is equal to 0.48. And remember that they have to be equal because the lysosome has to be in an isotonic solution. And they gave us that clue when they told us that they want to prevent swelling and lysis. So then it was like a complicated problem, and you had to know a lot of biology here to know or guess that this is what you had to do. But the math is really simple.
Problem 6. What is the pH of a solution that has a hydrogen ion concentration of A 1.45 times 10 to the negative 5th moles per liter? And the other two are the same. So what I did here was I added some formulas that all relate to pH. So anytime you get a pH problem, all you have to do is identify what they're giving you and what they're asking for. And then that's how you know which of these formulas to use. So the first one is pH plus POH is equal to 14. So if they give you a pH of 12 and they ask you what the POH is, you just do 14 minus 12 is equal to 2, and that's your POH. And then there's POH is equal to the negative log of the hydroxide ion concentration. And then you have pH is equal to the negative log of the hydrogen ion concentration. And then we have pH is equal to 14 minus the POH, and that's the same as the first one, just moved around. And then we have pH is equal to the pKa plus the log of the conjugate base over the conjugate acid. So in this question, they're saying, okay, they want to know the pH, and they're giving us the hydrogen ion concentration. So we know that we're going to go ahead and use this because the pH is equal to the negative log of the hydrogen ion concentration. So for A, let's see here, let's use red. Okay, so for A, all you do is pH is equal to the negative log of the hydrogen ion concentration. So here it is 1.45 times 10 to the negative 5th moles per liter, and that equals that equals 4.84. And make sure that you have a calculator that is able to do negative logs, because I started off with a calculator that didn't allow me to add the negative sign, and that was a hassle. So definitely I had to go buy another one. But I just went to a thrift store and I found one there for $2. But I think they have them at Walmart for like $15. So let me see if I can show you. So B, pH is equal to the negative log of 5.5 times 10 to the negative 10 moles per liter. That equals 9.26. If you're getting the same answers as me, that means that your calculator is fine. Okay, pH is equal to negative log of 2.0 times 10 to the negative 4. Okay, and that equals 3.70. Alright, it's that easy. Problem 7. What is the hydrogen ion concentration of a solution with pH of 2.52 and 4.89? So this is really easy. They're just giving us the pH this time, and we have to find the hydrogen ion concentration. So we just have to use this formula right here, and I'll help you set up problems A. So the hydrogen ion concentration is equal to 10 to the power of the pH, negative power of the pH. So for A, it is 2.52. So the hydrogen ion concentration equals, okay, so this equals 0.00301. So we have to convert it to scientific notation. So we go 1, 2, 3. So it's 3 point. Okay, there's actually a 9 over here. So it's 3.02 times 10 to the power of 1, 2, 3 negative 8. And then for B, the hydrogen ion concentration is equal to 10 to the negative 4.89, and that equals 0.000128. So 1, 2, 3, 4, 5. So the answer is 1.29 times 10 to the power of negative 1, 2, 3, 4, 5. And that's it.